The sodium-potassium ATPase consists of three main components. At its core lies the alpha subunit, the catalytic powerhouse. Then there's the beta subunit, essential for protein folding, cell adhesion, membrane localization, polarization, and possibly involved in potassium transport, mainly by its occlusion. It also modulates enzyme affinity to sodium and potassium ions. And then there's a lesser known participant, the FXYD protein family. One specific variant, FXYD2, found in renal cells is even referred to as gamma subunit. While not structurally essential, these FXYD proteins serve regulatory roles, subtly fine-tuning enzyme affinity for ligands, unlike the indispensable alpha and beta subunits. Sodium-potassium ATPase is called an enzyme because it catalyzes the hydrolysis of ATP. It is a pump because of its ability to move ions across membrane using energy. The pump operates by shifting between two major conformational states, E1 and E2. This cycling is driven by a sodium-dependent autophosphorylation and a potassium-dependent dephosphorylation. To simplify things, let's temporarily set aside the beta and gamma subunits and focus on the alpha subunit, where most of the action happens. The alpha subunit has a transmembrane domain that spans the membrane 10 times. Its cytoplasmic region houses three critical functional domains. One, the nucleotide binding domain, N domain, which exhibits kinase activity, contributing to the transfer of phosphate group from the ATP that binds to it. Apart from autophosphorylation, it plays a role in broader signaling pathways involving the sodium-potassium ATPase. 2. The phosphorylation P domain, where the actual phosphate transfer occurs. And 3. The actuator A domain, with phosphatase activity that helps in dephosphorylation. Now let's walk through the cycle. We begin at the E1 state where a segment of the transmembrane domain reorients to open an inner gate towards the cytoplasmic side. Magnesium ions and ATP binding to the N domain are critical here. At this stage, the negatively charged binding sites lined with aspartate and glutamate residues have a high affinity for sodium ions. Following the sodium binding, a phosphate group from ATP is transferred to the aspartate residue in the P domain, creating the E1P state. This phosphorylation event occludes the inner gate, trapping the bound sodium ions within the protein. Subsequent ADP release enables a conformational shift, opening the outer gate. Sodium is now released extracellularly, moving against its concentration gradient from lower to higher concentrations. This new conformation, the E2P state, now has a high affinity for potassium. Potassium ions bind from the extracellular side, along with a proton from the cytoplasmic side, which helps compensate for the charge imbalance and is also thought to facilitate sodium release. Potassium binding causes closure of the outer gate, and this triggers dephosphorylation of the P domain. As a fresh ATP molecule binds, the pump returns to the E1 conformation, reopening the inner gate. Now potassium ions are released into the cytoplasm and the pump is ready to start the cycle again, exchanging sodium for potassium across the membrane. When we talk about the functions of sodium-potassium ATPase, it maintains the cell volume by keeping out excess osmotic solutes like sodium. It maintains the membrane potential by creating a deficit of cation intracellularly. Remember, 3 sodium out and 2 potassium in. It provides a gradient for secondary active transport. It regulates cytoplasmic pH through sodium-hydrogen exchanger and calcium through sodium-calcium exchanger. It is involved in signal transduction for cell growth, differentiation, inflammation, muscle contractility, kidney function, and even behavior. Sodium-potassium ATPase oxidant amplification loop is postulated to be involved in aging, obesity, cardiovascular diseases through the generation of radical oxygen species. It's known to play a role in the pathogenesis of bipolar disorder. Altered behavior in animal models have also been observed. Ouabain is an endogenous cardiac glycoside that is synthesized in the adrenal and hypothalamus. The sodium-potassium ATPase acts as a receptor for this. It binds in the E2P form specifically. As a receptor for endogenous ouabain, it is involved in non-classical testosterone signaling pathway in Sertoli cells for spermatogenesis. It has a role in sperm motility. It affects natriuretic response to salt load. It is seen to have an association with primary hypertension. It modulates cell contact, several types of cell-cell interaction participating in polarization. hence a potential target of anti-cancer therapy due to its role in cell addition, motility, migration in cancer cells. Altered expression of enzyme subunits are also seen in various cancers. Since it's a mechanism of pathogenesis and normal functioning in some RNA and DNA virus, it can be a potential antiviral target by itself or as an adjunct. So you see, the enzyme goes far beyond what was traditionally taught to us. I leave you with a statement to ponder over. Hypokalemia potentiates digitalis toxicity. Rack your brains up and let me know the comments why. If you want more science-backed animation video to guide you through your medical journey, don't forget to hit that like button and subscribe. Keep learning.